Today, we're diving into how AI is helping us crack one of the biggest puzzles in human genetics, predicting the real-world impact of tiny variations in our DNA. We're going to break down some absolutely fascinating new research that has the power to change lives.
So I want you to imagine searching for a single misspelled word in a library that has millions of books. That is the exact challenge geneticists face every single day. We're on the hunt for a specific kind of typo called a missense variant. It's where one protein building block gets swapped for another. The thing is, every single one of us has millions of these genetic variants, so trying to pinpoint the one that's causing a rare disease, it's a monumental task.
And this isn't just some academic puzzle. For real families, this search can turn into a years-long, often heartbreaking journey from one specialist to the next, just trying to get an answer for their child's condition. And that journey, it has a name: the diagnostic odyssey.
All right, so let's frame this whole thing as what it is: a genetic detective problem. We have all the clues, the patient's entire genome, but how in the world do we find the culprit?
Well, here's the tough reality. Even with our most powerful gene sequencing technologies, the success rate for diagnosing some of these rare diseases can be as low as 25%. Let that sink in. That means for every 4 families searching for answers, 3 are left with no clear genetic explanation.
And this right here gets to the very heart of the challenge. Our older tools, well, they could tell us if a genetic variant might break a single protein. That's fine. But what they couldn't do was compare the impact across different proteins. They couldn't tell us if damaging protein A is, way more harmful than damaging protein B. They were missing that calibrated, system-wide view of our entire collection of proteins, what we call the proteome.
So, to solve this, researchers knew they needed a totally new approach. And it turns out the answer was in a powerful combination of data. It's really a tale of 2 very different but perfectly complementary datasets.
And this slide just perfectly lays out the strategy. On the left, you've got this super deep evolutionary data. We're talking about looking at genetic sequences across tons of species. This tells us which parts of our proteins have been protected for millions of years. In other words, the parts that are absolutely essential. And on the right, you have what's called population data. This is all the genetic variation we see in large, healthy human populations. Putting these 2 together, that is the key.
And this brings us to the new tool at the center of all this research, popEVE. It's what's known as a deep generative model. Think of it like a really sophisticated AI that learns the fundamental rules of protein language, and then it uses that knowledge to make predictions. It was designed from the ground up to weave together those 2 powerful data sources we just saw.
So how does it actually work? Well, it's basically a 3-step process. First up, popEVE learns from evolution. It analyzes sequences across all these species to figure out what's absolutely critical for a protein to do its job. Next, it calibrates with population data. It looks at which variants are commonly found in healthy humans and adjusts its understanding. The logic's pretty simple, right? If a variant is super common, it's probably not causing a severe disease. And finally, it generates one unified score. This allows us, really for the first time, to directly compare the potential danger of a variant in one gene to a variant in a completely different one.
Now, this is a really crucial point: fairness. Because popEVE uses a very simple measure from that population data, basically, is a variant seen or not seen, it manages to sidestep a huge problem other models have: ancestry bias. This makes its predictions much more equitable across diverse human populations, which is so important.
Okay, so the theory sounds amazing, but the big question is, does it actually work in practice? Let's take a look at the results.
This right here is a big one. A variant's ability to cause disease, its pathogenicity, it's not just a simple yes or no, it's a whole spectrum. And this graph shows that popEVE really gets that nuance. It gives clearly different scores to variants that cause severe childhood diseases versus those linked to more moderate adult-onset conditions. That level of detail is a massive step forward.
And here, you can see that separation in action. That purple curve, those are the popEVE scores for variants found in patients with severe disorders. The teal curve represents variants in healthy people. I mean, you can see that clear separation, right? popEVE is actually distinguishing the dangerous signals from all the harmless background noise. Now, this slide is incredibly important for anyone thinking about clinical use. Let's say you want to find 50% of the true disease-causing variants in a group of patients. To do that, popEVE only flags 11% of the healthy population as having a potentially severe variant.
A competing model, alpha missense, has to flag a whopping 44% of the population to find that same number of cases. This means popEVE generates way fewer false positives, which lets doctors focus on the candidates that are most likely to be the real answer.
And because it's so incredibly precise, it becomes this amazing discovery engine. In just one group of patients with severe developmental disorders, popEVE pointed to 123 new candidate genes that could be responsible for these conditions. That's huge.
Okay, let's tie this all together. What does this actually mean for cracking a patient's case?
This, right here, is a total game changer. Traditionally, to find a disease-causing variant in a child, you'd also have to sequence the parent's DNA to see what's new. But popEVE is so good at ranking how severe variants are that it can often pinpoint the likely cause from the child's genome alone. This is massive for what are called singleton cases, situations where, for whatever reason, you just don't have the parent's DNA.
And what's really cool is where these high-scoring variants actually end up. When you map them onto the 3D structures of proteins, they aren't just random. They cluster in these critical interaction sites, the exact places where proteins connect with other molecules to do their jobs. It's like they're landing right on the most critical parts of the machine, confirming the model is flagging variants with real, physical consequences.
When you get right down to it, it's all about finding that one causal variant. And in patients where we already know the disease-causing mutation, popEVE consistently ranks that specific variant as the single most dangerous one in that person's entire genome. It just rises right to the top of the list, telling the genetic detective exactly where to look.
So that leaves us with this incredible thought. Tools like popEVE are giving us a view of our own genetic blueprint that is clearer than anything we've ever had before. It really makes you wonder, doesn't it? With this powerful new lens, how many more of those heartbreaking diagnostic odysseys can we finally bring to an end? How many more mysteries are just waiting for us to solve them?